All right, so it's my great, thank you so much, Alexi, thank you so much for the introduction and the welcome. It's my great privilege and honor to be here as a guest professor. I really love the program and love the idea of interactions and fostering communication between different groups. And so far, we've been here with my family, and so we've been having a great blast. The city is really wonderful. My kids want all ready to move here, so things are going great. At some point, we'll have to take them back to the US, and that'll be challenging, but yeah, we'll survive. All right, so I'm an evolutionary biologist, and the connection with Alexis's team is through evolutionary biology. And so I always start by introducing ourselves and introducing the types of data we use and the kinds of questions we ask. And so evolutionary biology really starts with Darwin, and these are some of the types of data he used. He compared the anatomies of different organisms, he compared their embryologies, he looks extensively at the fossil record. It wasn't that rich, but he really thought deeply about it. And then he bred pigeons, lots of pigeons. And it's through these types of data and through these analyses that he came up with these sort of two foundational ideas that set up evolutionary biology as we know it today. One is that there is a mechanism for generating the diversity of life, and that mechanism is natural selection. And the other is that organisms are not specially created individually by a creator, but instead they're related through descent with modification from common ancestors. And much like Darwin sort of used these types of data, I think if he was around today, he would be really thrilled to have an additional data type. It's not that the other types of data are no good, but I think we have something really powerful, and that is the record of DNA, the sequences found in our genomes. So here is a quote from one of the pioneers of genomics, Eric Lander, who says the genome, it's a fossil record, it's a landscape, it's a whole geography of distributions, and it's a storybook that tells us the story of life so that you can read it like 1001 Arabian Nights and, find a different story in the genome every night. I don't know about, taking it to bed, as Eric suggests, but, you can certainly take it to the lab, and that's what my lab does. We analyze genomic data, we study the DNA record, and we try to understand evolution's patterns, evolution's processes, using computational and experimental approaches. And we're sort of strategically, or by design, if you like, I don't know how strategic it is, but we're sort of trying to be placed between sort of the folks that develop the software, the computational tools, and the folks that do the experiments. So we're somewhere in that middle zone. And so my lab does sort of a whole bunch of diverse things. We're interested in phylogenomics, and this is really what brings me to HITS and to Alexis's lab. A lot of the data that we use to do phylogenomics, and I'll explain what that is, rely on the use of fungal genomes. And so we have projects that focus on the evolutionary biology of budding yeast, or the evolutionary biology of some pathogenic fungi in a genus called Aspergillus. And then finally, we also work with mammalian genomes, and we're interested in the evolution of gestation length and mammalian pregnancy more generally. And so you can tell, right, these projects are really diverse, and the underlying glue is sort of the computational and experimental approaches that we use. They're shared between sort of the projects, and then the biology, of course, is very different for each one of these projects. Okay, and I'm also an evolutionary biologist living in Tennessee. For those of you who don't know, Tennessee is right under Kentucky and right above Alabama, and on the same meridian as Chicago, so southeastern United States. And for an evolutionary biology, Tennessee, sort of, especially in the US, is a special place. It's where about 100 years ago happened a really famous trial known as the Scopes monkey trial. Tennessee at that point in time had a law banning the teaching of evolution at schools, and one science teacher violated that law, taught science in high school, and was convicted. The charge was, or the conviction was reversed on a technicality, and so he never actually did time or paid a fine or anything like that. But a lot of the debates that we have about science and religion and the interaction between science and religion are founded sort of, or have their roots, at least in the US, in that trial. And the trial is sort of still, especially with what's going on in the US, is still very, very relevant today. And so part of my mission in the last few years, after sort of rising through the ranks, is to really try to unite evolutionary biologists on campus, and we have really diverse scholars studying evolution, and create a center focused on evolutionary studies. And so we have a website, we have activities, we're supporting graduate student training and so on and so forth. And we have a really nice logo that's inspired by the tree used in the book that Scopes used to teach evolution and get in trouble. So if you want to know more about that, I'm more than happy to talk to you anytime. And so what I want to talk to you about today is phylogenomics. Phylogenomics is really defined as the inference of evolutionary relationships among organisms using genome-scale amount of data. So typically, folks start with collecting tissues from different organisms or species or taxa. You'll hear me use these terms interchangeably. They sequence their genomes. Usually that sequencing happens in small bits. And so we have to develop methods to assemble the genomes and annotate them. And that's sort of usually the field of genomics. And then the phylo part, the evolutionary part, really starts once we have these genomes available. Then we have to identify the orthologs. We identify the gene, the corresponding gene from every species that has it, create multiple sequence alignments, and then analyze them using computational phylogenetics in order to try to come up with some hypothesis about species relationships. And as you'll see throughout my talk, for me, phylogeny is a really hypothesis of thinking about evolution, and they're not sort of given facts. We continuously revise them and so on. And phylogenetics is really a tool or an approach that has given us a lot of data to think about evolutionary relationships and how we go about them. So historically, as I said, right, Darwin started thinking about evolutionary relationships and phylogenies. He thought at the theoretical level that species have ancestors and descendants and so on, and they exist through time, but he didn't really do much. The first person who actually did something was in 1865, a scientist, if you can call him. He was a scientist at that point in time, St. George Jackson Mivart, and he took data from the vertebrae, the spinal vertebrae, the spinal column in the vertebrae of different mammals, and he built a phylogenetic tree of primates. So here is our species, Homo. I mean, it's close related to some other primates and more distantly related to some other primates. And then he did something interesting. He kept going and he analyzed data from the limbs of the same primates. And so in 1865, he came up with a different tree. And here Homo is in a different place. It's totally the outgroup of all the different primates. And so from the beginning, as soon as folks started using characters for building phylogenetic trees, they started seeing this conflict, this disagreement, and that this incongruence that I'm going to be talking about. Now, Darwin at that time was writing his book on the evolution of humans. This is the 1860s. And so he was reading Mivart's work, and he was interested, what tree should I use for my book that I'm writing? And so he wrote letters to Mivart asking about, which of the two trees is correct. And Mivart said, I have no idea, right? If you use the spinal column data, you get one hypothesis. If you use the skeleton, the appendicular skeleton, the appendages, you get a different tree. Good luck. And that's pretty much the state of the field for the next 100 years. People use morphological data for different groups, and depending on with which expert you trained, you got different beliefs or sets of beliefs, and folks argued. By the late 1800s, there were huge disagreements, and it's fair to say that while people believe that there are evolutionary relationships, they didn't believe they had the tools to infer them. And the situation really didn't change until the revolution in molecular biology. That's when we start having DNA sequences and protein sequences. At the beginning, very scant amounts of DNA and protein sequences. But that's where the first ideas come that, hey, maybe we can use these molecules that contain information about our function as well as our ancestry to infer phylogenetic trees. And that's what they did. So pretty quickly in the 1960s onwards, people start building phylogenetic trees with different types of molecular data. In the beginning, it was using allele frequencies from different populations of humans. Here is a famous phylogenetic tree by Luca Cavalli-Sforza, a pioneer in human population genetics, and Anthony Edwards. And then arguably sort of the most famous tree from that sort of first period is a tree built on 16S RNA that identified that there is a separate kingdom of life called archaea, and that's distinct from bacteria and eukaryotes. And that was in 1977. I'm truncating a lot of evolutionary history, or a lot of history of evolutionary biology, in very few slides. But it's fair to say that, until the late 1970s, 1980s, there was this idea that the trees we built from gene sequence data or protein sequence data are equivalent to the species tree. There is really no conceptual distinction about them. As you'll see in my talk, I want to argue that that's definitely not the case, that gene trees are very different from species trees, and that has major implications of how we study phylogenomics. And the reason that gene trees and species trees are not the same comes down to two sets of factors that we've labeled analytical and biological. So analytical have to do with things like stochastic error from sampling, and systematic error when your data don't fit the model that you're using. And the biological factors have to do with the biology of genes. And there are many processes, evolutionary processes, that can cause the histories of genes to deviate from the species history, and these result in disagreements between gene trees and species trees. So let me give you some very brief examples of some of these. I don't have the time to be exhaustive. Sampling error. Here's an alignment that I think I took from Alexis, one of Alexis' practicals back in time. 10 different species of vertebrates, 50 amino acids aligned. I think it's a hemoglobin or something like that. Really quick data set, not the whole gene. If you build a quick phylogeny out of this, you see that humans group with chickens and they're far away from the other mammals. Of course, that doesn't mean we're birds. It just means that we sampled too little of the sequence. Nowadays, sampling error is not so big an issue because we have genomic data, and I'll show you how we've come to that conclusion. But nevertheless, when we analyze individual genes, that potential is always a problem. Systematic error is a little bit more challenging to deal with. The classic example of systematic error in the field of phylogenetics comes from simulation studies using this very simple 4-taxon tree. So we have 4 taxa A, B, C, and D, and we set the branch lengths of these 3 branches to be at length Q, and these 2 long branches at length P. And as we make Q smaller and P bigger, we enter an area where the more data we add to the inference, no matter what method you use or algorithm, your results are inconsistent in that they give you the wrong topology, right? So you'll simulate data under the hypothesis that A and C, have an internode in between them of length Q. But what you'll end up doing if you infer the phylogeny is that A and C are sister to each other and there is no internode. Okay, so that's sort of the area of non-consistency. And so that's problematic because that means that, right, the more you add data, the more confident you become in the wrong solution. And that's a big issue in phylogenetics nowadays. And later on, I'll show you an example of its action in actual data set. So that has come to be known as long branch attraction because these 2 long branches, as they get longer, they begin to attract, and our inference reflects that. Okay. Now, biological factors have to do with, as I said, the histories of genes being different. There are many reasons why the histories of genes could be different from the species carrying them. One of the most common is a process that's called lineage sorting. So what I'm depicting here is the histories of 3 species, A, B, and C, and these are depicted by sort of the big barrels. And each individual in the population is depicted like as a little dot. And so you have, right, these individuals existed some generations in the past, these are the present individuals, and so on. And so typically when we infer phylogenetic trees, we go and pick, right, one individual, one allele from each one of these species, and we built our phylogeny. And so if you look at this tree, right, we have the speciation of species A from the common ancestor of species B and C at this point in time, right? That's when the 2 lineages diverge. And then another speciation of lineages B and C a little bit later in time in a short succession. But if you track the histories of these 3 alleles that we've sampled from the population, the way I've drawn it, what you note is that the splitting of the alleles is different. The history of the alleles is different than the history of the species. Here, the C allele split from AB before. It's split from in a different manner, right? So here we have speciation of A from B and C. Here we have speciation of C from A and B. And then later on, we have another splitting of the A and B alleles. And so the topology that we get from species is that B and C are each other's closest relatives, whereas the topology that we get from the allele histories is that A and B are sister to each other. A and B alleles are sister to each other, and C is the outgroup. So that happens, relatively commonly, especially when the time between speciation events is very short. The classical example of lineage sorting is our own species. So if we look at the human genome and compare it with the chimp and gorilla genomes, and we ask at every site in the genome how many are shared with chimpanzee and indicate that chimp is our most close relative, and how many indicate one of the other two possible histories, that human and gorilla are each other's closest relatives, or that chimp and gorilla are each other's closest relatives and humans is the outgroup. What you find is about 80% of the genome suggests that humans and chimps at the molecular level were each other's closest relatives. But then for about 10% of alleles, a different hypothesis is supported. So for about 10% of alleles, the allele that we have in our genomes is more close related to that of the gorilla than to chimp. And then for another 10% or so, the gorilla allele is most close related to that of chimp, and humans are more distantly related. Okay, so that's the process of lineage sorting. Gene duplication loss happens sort of through a similar process, if you like. We have an ancestral duplication of a gene and creates two copies in an ancestral genome. As species diversify, they lose different copies of that duplicated allele. And that means that you can sample genes that are paralogs of each other and not orthologs. So the evolutionary history of the genes you're sampling reflects the history of the duplication of the genes rather than the history of the species carrying them. And then the final process that I want to mention, I'm skipping a few, is the process of hybridization and introgression. That's when you have species that mix the genomes and create hybrids. Sometimes these hybrids are rather successful. Here is a phylogeny of the genus Saccharomyces, the baker's yeast, Saccharomyces cerevisiae, which is the cornerstone of the baking, brewing, and wine industries, and some of its relatives. And there is a very rich history of introgression and hybridization that we're aware of until we've discovered the parental species, and that happened in some cases relatively recently. We thought, some of the species that are actually hybrids were actually not hybrids, and so it can be hard to detect. And then also some species, some of these hybrid species have become economically very important. The cornerstone of the lager brewing industries is Saccharomyces pastorianus, which is a hybrid between Saccharomyces cerevisiae and Eubayanus. Cerevisiae likes warm temperatures. British English ales are brewed with Saccharomyces cerevisiae only. Saccharomyces eubayanus likes to live in Patagonia on high altitude forests. The hybridization gave cerevisiae the ability to grow in the cold. And so pastorianus has the trait of, or has the ability to make really beautiful ethanol and really nice and tasty beer at cold temperatures, which was the trait conferred by Eubayanus. And so that can be really interesting and really informative and evolutionary really interesting. But for the context of phylogenetics, this really confuses the things. So I hope I've convinced you that, right, studying histories of genes and comparing them to histories of species is interesting, but the two are conceptually distinct. At this point, you might be wondering, what does this have to do with phylogenomics and genomic amounts of data? You may have some inklings, but it basically comes down to, the title of my talk, that all this sort of disagreement or differences between gene trees and species trees manifests itself as incongruence. And so simply put, if I have a gene or I do a particular analysis from taxa A, B, and C, then I get one topology that, right, A and B are sister to each other and C is the outgroup. And then I want to be thorough and I go and collect another gene or do a different type of analysis and I get, a different result that A and C are sister to each other and B is the outgroup. That makes me uncertain about the species phylogeny, and that conflict, that incongruence, that disagreement is is potentially really interesting. And so back in the day when I was a postdoc, this were the early days of genomics. So the human genome was sequenced in 2001. I started my postdoc, or was finished sequencing in 2001. I started my postdoc in 2002. And so the first genomes of multiple species from the same lineage were coming out, and they happened to be Saccharomyces and relatives, the budding yeast that we're still working on. And so back then, we wanted to systematically evaluate this level of incongruence and see if it really is the case with genome scale amounts of data. And so we collected a data set of about 100 genes from these 8 species, a rather basic data set nowadays, but back then it was really big and required a lot of effort. And what we saw is that, for every gene, and yeast genes are not very imaginatively named, but every gene was basically showing us a different phylogeny. And so if you look at this gene, YBL0921C, and compare it to YBL031W, you'll see that the positions of these 2 species, Castellii and Cloveri, have been switched. If you compare this gene with this gene, you see that relationships among the close related species next to Cerevisiae are altered, and so on and so forth. And so we saw about 30 different topologies among a set of 100 gene trees, which was really surprising at the time. In retrospect, it shouldn't have been surprising, but back then it really surprised people. And it caused one of the reviewers of our study to say, plainly stated, taxonomies keep digging the same hole for themselves and falling down it. All that has changed over the years is the sophistication of the shovel. Really, really inspiring words to receive as a postdoc. Although I'm grateful, this reviewer was actually positive for our work, because when we concatenated, we analyzed all the individual genes as a single super gene, if you like, as a single locus. We got one topology that was significantly more strongly supported than all others, irrespective of method, irrespective of data type, and suggested that this is the relationship of these 8 species. That has held true still to this day, which is very nice. And more importantly, it sort of gave us way to sort of start asking questions about, how do we get confident about evolutionary relationships? And so clearly 100 genes was more than enough to robustly resolve the phylogeny of these 8 species. But what we could do is to go and say, well, for any particular branch, how many genes do I really need to start getting confidence that this is the true relationships of these species? And so we could, pick a gene at a time randomly, or 2 genes, or 3 genes, or 4 genes, and so on, and measure some degree of confidence. In our case, it was done with bootstrapping, of how confident we became in that relationship being resolved. And one of the conclusions of our study is that for this particular tree, you needed about 20 genes to feel confident that your phylogeny was robust. So there was a commentary written actually by an editor in Nature, Henry G., who called this the beginning of the end of incongruence, that now that we have genome scale amounts of data, incongruence would disappear. I wish that was the case, although I might not have a job if that was the case. But nevertheless, it started sort of a whole bunch of studies that use this approach of using lots of genes to build phylogenomic trees. And the key word that I want you to remember here is people talk about resolving these branches. But what happened as these studies started coming out is that, have we eliminated incongruence? In fact, we didn't, because we started seeing lots of disagreements. So I want to here separate sort of 2 parts as we look into phylogenetics to inform, the history of evolution. I want to distinguish events that happen in the recent past and trying to infer evolutionary relationships among close related species, or even populations of a particular species, from very distantly related, trying to resolve relationships of very distantly related groups of organisms. So if we look in the recent past, so say evolution of human populations, things are straightforward. There is not much systematic errors. Our models work well. We have lots of data. We have lots of samples and so on. And so we can infer pretty accurately that, we hybridize with Neanderthals, and some of our genes in our genomes come from Neanderthals. We can even infer unknown hominins that have contributed alleles or variation in our genomes or in the genomes of some human populations and so on. So our accuracy is pretty good. If we go a little bit deeper, so say relationships of different primate genera, such as these 4 genera, there is Nomascus, there is Hoolock, Symphalangus, and 2 species of Hylobatus. These 4 groups, right, have 4 possible unrooted trees. You can root the tree in 5 of the different branches of the tree. So there's a total of 15 topologies. And so if you go to their genomes and say, what % of the genome supports this topology, you get a picture that looks like this, right? So 15% of the genome supports this phylogeny, 13% supports this phylogeny, 10% this, and so on and so forth, right? So what is the phylogeny of these species? I don't know. What I know is, right, they've hybridized, they've introgressed such that there is no single species tree that accurately reflects their evolutionary history. And so, but we know that that's the case, right? We have the tools, we have the models to figure out that that's the case. At deeper levels, incongruence is much more challenging because all we see is disagreement between phylogenies, but it's much harder to find the factors that contribute to it. And so I'll give you just a couple of examples where that's the case and highlight how controversial it is. So relationships between gastropods, snails and the like, bivalves, mussels and the like, and scaphopods. So a paper in Nature in 2011 finds that gastropods and bivalves are sister to each other, more close related. Another paper 3 months after, also in Nature, different relationship. I can't tell, these 2 studies were state-of-the-art at the time, used phylogenomic data, state-of-the-art methods. There is nothing wrong with either of them, but they significantly and very strongly disagree on this topology. Another relationship that has become, for the last 10 years, has become very, very controversial, concerns who is the sister group to the rest of animals. So we have bilaterally symmetrical animals, right, that we have an axis of symmetry. That's us, that's insects, that's most of the animals you know. And then there are ctenophores. This is a small group of pelagic organisms called comb jellies, and then sponges. And over the years, there has been a lot of debate on whether sponges are the sister group to the rest of animals, or whether it's ctenophores who are the sister group and sponges group with the rest. And these 2 studies appeared within a day of each other in PNAS. They even have a shared author in between the 2 studies. So that author is really confused as to what the relationships are at the base of the animal tree. Okay, so then part of our research programs is figuring out why are these branches incongruent, and how can we develop methods to resolve them. Part of the answer, at least at a certain level, is that they are incongruent because every gene that we sample for these data sets seems to be disagreeing with the topology or the tree that every other gene is giving. So if we look in a plant data set, about 182 of 184 genes produce topologies that differ from the other genes. For mammals, that's 440 out of 447. For yeasts, a data set that we generated some time ago, all 1,000 plus genes differ from each other in terms of topology. Same for birds and so on and so forth. So there is lots of disagreement at the gene level. And so part of what I've been doing is sort of trying to develop methods that enable us to capture that disagreement and see which disagreement is meaningful and which simply reflects lack of information. So let me give you an example from our yeast work. So this is a phylogeny of a group of yeasts, and what I've depicted here in each one of the branches is what we call gene support frequency. So the percentage of single gene trees that support a particular relationship. And so for this branch, you can see 95% of our 1,070 gene trees support this branch. I would probably bet my house that that branch is correct. There's no way that branch is incorrect in its inference. But you also have these branches here that's supported by only 29%, 30% of gene trees. And so here it really matters to figure out, what the other 71% of gene trees say. If the other 71% says, I really have no opinion, I'm totally uninformative, then that 30% support is pretty good, and you can make something out of it. If the other 30% if the other 70% is telling you something very different and strongly, supporting something very different, then of course your conclusion would be different. And so we've been trying to take advantage of these types of data to quantify incongruence and to think about how we handle incongruence in phylogenomic data sets. And I want to mention just a couple of these methods: internode certainty that was developed together with Alexis's team, and then some efforts in trying to identify the phylogenetic signal of single genes. And so internode certainty, these are the papers, or some of the papers that we've generated with Alexis' team. Basically takes advantage, it's a measure of support for a given internode that takes into account its frequency in a particular set of trees, and also takes into account the frequency of the most prevalent conflicting internode in that same set of trees. And so if you have two alternative hypotheses that are supported each by 50% of your gene trees, or each by 30% of your gene trees, then when you calculate the certainty, it's going to be it's going to be zero, right? So if the ratio of support for two conflicting internodes is half, then your certainty will be minimized. If the ratio of support is in favor of one or the other topology, the other relationship, then your certainty will be maximized. And that's the point, right? So you measure the conflict. You can calculate the same concept for the entire tree by summing all the internode certainties across a phylogeny, and then calculate that. So let me give you an example of how that's useful. It takes us further from what a simple gene support frequency would give us. And so here are two branches, 62 and 52, gene support frequencies. So this branch is supported by 62% of genes. This branch is supported by 52%, right? At face value, we would say, well, these are very, very similar. You wouldn't have a strong opinion for the validity of one over the other, or the truth of one over the other. But then by calculating internode certainty, we find that the most prevalent conflicting internode has a frequency of 6, right? So you have this relationship supported by 62% of your trees, and this next most prevalent conflict is by only 6%. And so you get a relatively high internode certainty value. In contrast, for the branch that's 52, the support for the most prevalent conflicting internode is 29%, so much more significant conflict. The internode certainty value is very low. We know from prior work that there is a history of hybridization in these lineages, as I was telling you about. And so that 29% reflects actually, right, that signal of hybridization and introgression. And so internode certainty is able to pick that up, whereas gene support frequency would not. The other approach that we've developed is relying on quantifying phylogenetic signal. What do I mean by that? So we go back to the major debate about what is the sister group to the rest of animals. Is it comb jellies, ctenophores, or sponges, poriferans? And so we can take any particular data set, any particular gene in a data set, and given this topology and given a particular model of sequence evolution, calculate the probability of the sequence alignment fitting that, right? So we can get a likelihood score under this hypothesis, and then we can get a likelihood score under the alternative hypothesis that sponges are the sister group, okay? And we'll call phylogenetic signals simply the difference in the log likelihood scores of the two hypotheses, right? So same alignment, same models and so on, we alter the trees and we ask, which one has the better likelihood fit, and that becomes the phylogenetic signal for, their difference becomes the phylogenetic signal for one or the other hypothesis. And so if we do that in the context of a phylogenetic matrix, so we can plot phylogenetic signal on the y-axis, arrange the genes individually on the x-axis, and this is a data set of 1,080 genes from 36 animals. And so the picture looks like this. And so anything above 0 supports topology 1, that comb jellies are the sister group to the rest of animals, and anything below 0 supports sponges as a sister group. Okay, so it gives us a visual idea of what is the support for this. Now you can sort them from the highest to the lowest value, and this is the same graph but sorted. And so the area under the curve above and below 0 basically gives you the amount of support for ctenophores as the sister group or sponges as the sister group. So visually helps biologists identify, the relative support for their two alternative hypotheses, of course, under a particular model of sequence evolution. So you can apply that not for the xenophore debate, but for every debate you can think of in phylogenomics. There are quite a few. And so we analyze several different plant data sets, several different vertebrate and yeast data sets, trying to identify these branches that have been controversial in the literature. And these are the plots of the phylogenetic signal on a gene by gene basis. And what you can tell, probably you can't tell because the graph is too small, but there are some hypotheses, you have to take my word for it, there are some controversies where it clearly seems that if you account for phylogenetic signal, the support for one hypothesis of the other is pretty strong. And so you could think that, this method helps you sort of go towards the correct inference of relationships. But then there are these other relationships, and that you can't see, where there is a single gene that seems to be having really strong signal, and all the others seem to have almost no opinion, right? So there is this contrast. And so let me give you an example of each. So this is a case where the signal is really strong in favor of one hypothesis over the other. So this is the debate about whether turtles are the sister group to birds and crocodiles or just to crocodiles. It seems like the first hypothesis is the one that's most supported by the current data that we have. And so clearly very different evolutionary signals. And here is a case from the yeast that concerns two topologies, and it really comes down to the placement of this group called Ascoidaceae. It's a single taxon that we have, and it can be placed either in the middle of this phylogeny or as an outgroup of this phylogeny. And the phylogenetic signal is exactly the same, except for that really long. There is one gene that has extraordinary strong signal for topology 1 over topology 2. Let me show the same graph to you, unsorted. And so that's the really strong gene. So again, right, so everything above 0 supports topology 1, everything below 0 supports topology 2. The question we had was, well, what if you remove that gene, right? So you have a phylogenetic data matrix, how much does that influence? And so if we go and remove this gene and build a phylogeny instead of 1,233 genes, phylogeny of 1,232 genes from the same set of taxa, what you do is you switch support. Instead of topology 1, your concatenation analysis now supports topology 2. So a single gene removed in a phylogeny changes the topology in just that branch. The rest of the topology doesn't change much, but that particular gene seems to have all the signal for relationships between your groups. And so what's going on? Obviously, we talked about some biological factors that may be accounting for that. There is also the hypothesis that this is due to analytical factors. In the case of the yeast relationships and that controversy with that single genes, we're in a good position because part of the other side of my group, together with collaborators, what we've been doing is building a bigger and ever bigger and bigger data set of budding yeast genomes. And so our goal is to sequence all known budding yeast genomes, and I think we've achieved that. So around 2018, we had a data set where we now had 330 or so budding yeast species, much larger set of genes, 2,400. And we can plot the phylogeny and calculate similarly sort of areas of the phylogeny where there's disagreement between different methods. And we knew that our gene that caused the trouble, that gave this really strong signal for one topology over the other, was also included in this data matrix. And we knew that the relationship, the controversy between topology 1 and topology 2 was no longer present here. Seemed like it was always topology 1 we were getting. We never saw topology 2 in any of our analysis with different parameters, different settings, and so on and so forth. And so what has changed in our data matrix, apart from increasing the number of genes and increasing the number of taxa, was specifically that we had added 4 taxa now instead of 1. So our original analysis of the 86 species included just Ascoidea rubescens, and it was its placement that was the problem. Now we had also Asiatica and these 2 Saccharomycopsis species. And so our hypothesis was that maybe what was happening in the original analysis was systematic error. There was a long branch, and that was being attracted to the rest of the phylogeny, and that was creating the issue and the controversy was making that gene misguided. So if that was the case, we can do an experiment, and we did it. So we can calculate the phylogenetic signal for that particular gene that gave us the trouble. If we only include Ascoidea rubescens, so we have just one taxon in our phylogenetic analysis or phylogenomic data matrix, or all 4 taxa, so Ascoidea and the other 3 relatives. And we can plot the phylogenetic signal for that gene as well as for every other gene in our data matrix. Okay, and so if we do that, this is the picture. And so this is the phylogenetic signal for when we have the entire data matrix. This is the phylogenetic signal when we've removed these 3 taxa, and this is our favorite gene, right? So very, very strong phylogenetic signal if we have just one taxon included. But then if we have 4 taxa included, it's still very strong, but not crazy high. And so that indicates that as we add taxa, we're probably better able to estimate the evolution of that species and place it better on the phylogeny. And so that's encouraging because, we're sequencing more and more genomes, we're generating bigger and bigger data matrices. It's nice to see that at least for some problems or some controversies go away simply by adding additional data. And so the way forward, as I wrap up, it used to be, I think, that phylogenetics was, we generated multiple sequence alignments, we built data matrices, and then we applied different optimality criteria, likelihood Bayesian inference, different approaches. I've talked about concatenation. There is another one that relies on making use of the individual gene trees called coalescence, and then stopping there and saying, this is the phylogeny. And I think we've entered an era where we really have to assess what's going on under the hood. We have lots and lots of data, but these data disagree with each other often or have alternative hypotheses. And we really have to change our mindset instead of saying, this is the new phylogeny and it's fully resolved, to really report what branches of the phylogeny we're really confident about. And we would bet, our houses, maybe not our houses, but we would bet some money or some beers. And then others that, we're really not certain about. And I think that would, take a lot of the debates or the strong disagreements on, particular favorite phylogenetic hypothesis away from the literature. Now, as we move forward, we're building even larger data matrices. So here is a phylogeny from last year that we built, phylogeny of the entire kingdom of fungi. So it has 1,600 species, genome scale amounts of data. We were able to find 290 or so genes for that data matrix. And so the tasks are getting bigger. And so that means that our challenges are changing in nature, so to speak, in that as we build ever bigger, or as we try to infer bigger and bigger trees, I think you're all aware, having Alexis and his group here, that means that the number of possible topologies is increasing quite dramatically. So even for a set of species like 50, the number of possible unrooted bifurcating trees is astronomically or more than astronomically high. If you go to 1,600 or 1,700 or thousands or millions, it's getting obviously even bigger. And so how do we efficiently search that space? I don't know, but that's a challenge. And then how do we deal with the speed and accuracy? So obviously, analyzing data matrices like these takes a lot of time, months on supercompute clusters. And so at some point, we're going to reach data set sizes where we'll have to take out taxa if we want to, our postdocs to find faculty positions and get papers and so on. And so we've done some analysis trying to assess speed and accuracy of phylogenomic software. And it's very clear there's a trade-off. And so if we look at software like FastTree, it's fast, but its accuracy is really low. And if we look at software like RexML and IQTree, the state-of-the-art software, they're accurate, but they're slower. And so how we deal with that trade-off, I think, is going to be a really, really important issue for the days to come. And then the other thing we've sort of noticed, and Alexis' team had come up with sort of similar conclusions a little bit before us, is that, and this really originated by just trying to reanalyze our data set. And one of my postdocs, Xingjing Shen, found that he couldn't replicate some of his analysis. So he was using exactly the same alignments to infer phylogenies of some of the genes that he knew were giving particular topologies. And when he rerun it on the cluster, exactly the same seed number and some parameters, he was getting different results. And originally we thought something was messed up, but he tried with different data sets. He tried it systematically, and we found that if you use state-of-the-art software, somewhere between 9 and 20%, 10 and 20% of your gene trees are reproducible. That is, if you run it with exactly the same parameters, you'll find differences. Now, the good news is that these are not just random gene trees. They are the genes that tend to be less informative, tend to have lower amounts of phylogenetic signal. And so if you do simulation studies where you plot the phylogenetic informativeness, how much phylogenetic signal you have in a particular gene, and what is the percentage of irreproducible gene trees that you find, they really correlate with lack of information, which makes a lot of sense. The optimizations that these softwares have makes them, it gives rise to the reproducibility. And so, so these are some of the challenges. I don't want to end on a note about what are the challenges ahead. So I mentioned that we're working to try and generate even bigger data sets. Probably the most exciting data set that we have is one where we've sequenced every known species of an entire subphylum of budding yeast. And so we have, the nice thing about budding yeast is that you can keep them in the lab, and we have the type strains of almost every species, and we've sequenced them, and this is the phylogeny of the 1,200 or so species in their different clades. And so that's what we'll be analyzing in the next few months. So I'll stop there by acknowledging the people that did the work. A lot of the phylogenomic work in the early days was done, including the work on internode certainty, was done by Leonidas Salihos. He's now faculty at the New York Institute of Technology, and that was done partly in collaboration with Sarah Luttertop in Alexis's group. We're grateful for all that. And then the phylogenomic work on the yeast and phylogenetic signal was done by Jingjing Shen, often in close collaboration with Xiao Fan. Xiao Fan was also involved in the later stages of development of the internode certainty methods. And I talked a lot about the yeast data sets, not so much for the discoveries we've done about evolution of their metabolism, but a lot of that work has been generated in collaboration with Chris Hittinger's lab at the University of Wisconsin-Madison and Mariseth Gronwald at the Westerdyke Fungal Biodiversity Institute. And then finally, the funding for most of the phylogenetics work has come from NSF, but also we get funding from NIH and Barr's Wellcome Fund and a few other organizations. So thank you so much for having me for the next few months. And thank you so much for giving me the opportunity to tell you about our work. I'll be glad to take any questions you may have. Thanks.